I am Gerhard Leinenga from the University of Queensland, and I will be presenting our findings on the transcriptional signature in microglia isolated from an Alzheimer's disease model treated with focused ultrasound-mediated blood-brain barrier opening. In the Gertz lab, we are interested in how we might use focused ultrasound to treat Alzheimer's disease, or AD. We use focused ultrasound with microbubbles to disrupt the blood-brain barrier, and we are studying how doing this reduces amyloid plaques in the brain and improves cognition. AD is a common neurodegenerative disease characterized by two neuropathological hallmarks, an extracellular plaque pathology composed of amyloid beta peptides, and intracellular neurofibrillary tangles, which are made up of tau protein. These increase in the brain with increasing severity of dementia symptoms in Alzheimer's disease. Barriers in the brain, such as the blood-brain barrier, maintain homeostasis, and disrupting it with focused ultrasound can be used to deliver drugs, but we are also interested in the cell type specific bioeffects that disrupting the blood-brain barrier has. A cell type we are particularly interested in are microglia. These are the resident immune cells and phagocytes in the brain. They have numerous functions that have been elucidated in many recent papers, and also they're believed to be key players in the pathology of Alzheimer's disease, as, for example, microglia-specific genes increase the risk of Alzheimer's disease, such as TREM2. So our approach to open the blood-brain barrier using focused ultrasound involves injecting in-house prepared microbubbles and then treating many spots with focused ultrasound to cover the whole brain of a mouse, with short sonication durations in each spot, which results in blood-brain barrier opening throughout the entire brain, as you can see with Evans blue staining, without causing any observable damage to the brain. We called this scanning ultrasound, or SUS. We have previously shown that applying SUS treatment reduces the amyloid plaque levels in AD model mice. So the model we use here is APP23 mice, and this was published previously. We have recently compared in this paper the effects of SUS to the effects of aducanumab. So aducanumab is an anti-A beta antibody that showed efficacy in clearing plaques in human patients in phase 3 clinical trials. And what we find is that the plaque clearing effect of SUS was similar to the low-dose aducanumab treatment. And the combination of SUS plus aducanumab cleared plaques more effectively than either aducanumab alone or SUS. So we believe that the mechanisms of plaque clearance is through increased microglial phagocytosis of amyloid beta, which we have shown in previous studies by staining sections for amyloid beta plaques and microglia, and showing that after SUS, microglia contain more plaques in their lysosomal compartments, quantified here as the A-beta internalization ratio. The increased plaque phagocytosis occurred at the same time as there were pronounced changes to the microglia morphology. So the microglia here are more rounded up and phagocytic looking, and they contain more lysosomal markers as well. Shown here is percentage CD68 area. We have also shown in aged AD mice that the number of microglia surrounding plaques increases after SUS treatment, as shown in the study where the number of microglia around plaques and the size of plaques was counted, showing a pronounced increase of the number of microglia per plaque normalized to the area of individual plaques. So to gain a finer and molecular understanding of how microglia are altered by SUS in AD model mice, we performed RNA sequencing to obtain transcriptomic profiles specifically from microglial cells in aged AD mice that were treated with SUS or sham in this case being anesthetic and microbubbles and being placed under the ultrasound transducer without it being turned on. And we analyzed mice and collected microglia from their brains 2 days after one SUS treatment. So here in this scheme of our experiment, it shows that we isolated microglia and sorted them into two populations using flow cytometry based on whether they were actively phagocytosing amyloid or not. And whether they were actively phagocytosing amyloid plaques was determined by the presence inside the microglia of the plaque-binding dye methoxyXO4, which was injected 2 hours prior to sacrifice. And we separated these populations and performed bulk RNA sequencing on these plaque-positive and plaque-negative microglial populations. So principal component analysis and hierarchical clustering analysis revealed that there's a clear segregation between the samples comparing the APP23 or AD model mice to wild-type mice. And this bioinformatic analysis segregates distinct microglial populations based on their methoxyXO4 status, so whether they have phagocytosed amyloid plaques or not, as well as the treatment. So the SUS treatment and the sham-treated animals have marked differences in their transcriptomic profile in the AD model mice samples. So our analysis identified 397 differentially enriched genes, with 155 genes being specific for XO4 positive cells, 199 genes specific for XO4 negative microglial cells, and 123 genes being differentially regulated independent of whether the microglial cells contained amyloid plaques or not. Analyzing the treatment dependency patterns, we observed that most of the upregulated genes were induced by SUS, with a total of 353 enriched genes across all the A-beta internalization levels, and only 44 genes that were down-regulated following the SUS treatment. Volcano plots identifying the transcripts that were upregulated by the SUS treatment in the XO4 positive and XO4 negative microglial cells are shown here. The functionally enriched pathways induced by SUS treatment are shown here for a gene ontology analysis. And this reveals that cell cycle, DNA replication, and DNA metabolic processes are the most enriched terms in this analysis. Similarly, KEGG pathway analysis reveals that the top functionally enriched pathways are cell cycle, as well as phagosome, which is the cellular machinery for affecting phagocytosis, which we are interested in very much. And so here we've plotted the significantly altered genes in the phagosome pathway on the left for both XO4 negative microglia and XO4 positive microglia, and on the right, the significantly altered genes in the cell cycle pathway are shown for both XO4 negative and XO4 positive microglia. So in conclusion, we have shown that blood-brain barrier opening by focused ultrasound and microbubbles alters the transcriptional profile, the morphology, and the phagocytic ability of microglia, and also results in microglial proliferation. We believe that these transcriptional changes in microglia may be of significant importance in the effect of amyloid clearance after ultrasound blood-brain barrier opening, and also the memory improvement that we observe in AD model mice treated with focused ultrasound blood-brain barrier opening. And I would like to acknowledge all the people in the Gertz lab who helped with this work, as well as our collaborators who helped with this work from Monash University. Thank you.